So we've been looking at the DNA contamination inside vials of messenger RNA vaccine, and we've seen that it's often very high. These are the vials tested by Dr. David Speichier in Canada. This is the maximum legal amount of DNA in the vaccines. It's 10 nanograms per dose. But we see that all of the vials he tested are way above that. And in this Australian vial, it's very, very high, both spike protein and this original DNA in the plasmid here, and this SV40 promoter, which we've looked at as being particularly dangerous as a cancer risk. But the point is, this level of 10 nanograms per mil was designed to prevent contamination with naked DNA. And I want to show you what I mean, because this is a really important distinction. So here we have a human cell. Let's imagine this is a human cell here. That's the cell, that's the nucleus, and that's some of our own DNA inside it. Now, if some synthetic DNA is injected, there's a risk that that DNA can get into the human cell, which is what we don't want. But this DNA in itself is water soluble, whereas the membranes that go round about the outside of the cell here, this cell membrane is fatty. So it's hard for the naked DNA to get in. It's called a phospholipid bilayer. It's a fatty layer. So 10 nanograms per dose of DNA there, you're going to get very little going into the cell. You're going to get very little transfection, because the DNA is basically, you'll get a tiny bit going in, but virtually none, because most of the DNA gets to the membrane, can't get in. But the problem is, with these mRNA vaccines, the DNA is not naked. No, it is clothed. And it's clothed in a lipid nanoparticle like that. Now, the key thing about this lipid nanoparticle is that this can link up to the cell membrane here, like that, because they're both lipids. So the two can simply come together. The two membranes, the cell membrane and the lipid nanoparticle membrane, will simply fuse together, just like two bubbles when you're blowing bubbles with children. They'll just fuse together. And once they're fused together, that means that the DNA can very readily just get into the cell. You can just transfect into the cell. So you'll end up with this E. coli DNA with its spike protein DNA and its SV40 promoter, which, as we've said, will inhibit the body's own P53 tumour suppressor gene, making cancer more likely. And David's also told us that the SV40 will make it even more likely. The presence of the SV40 sequence in the DNA will make it even more likely that this DNA gets into our own DNA here and could potentially contaminate our own DNA, which is more of a concern. So we could argue about these guidelines being dangerously high already, but the guidelines were designed for naked DNA, which is going to find it very hard to get into the cells, whereas now we're using it to adjudicate the amount of DNA that's wrapped in the lipid nanoparticles. And the factor here is 100. It's 100. So it is 100 times easier for the DNA that's in a lipid nanoparticle to get into the cell than it is the naked DNA. So I guess that means it's reasonable to assume that if the DNA is in a lipid nanoparticle, the amount of DNA getting into the cell will be 100 times more than were the DNA naked. Therefore, I feel these guidelines should be changed and reduced by a factor of at least 100 to allow for the fact that the DNA in the lipid nanoparticle will have much greater cellular transfection than the naked DNA. And even with that, we see that, I mean, these levels are just way above the legal amount, but as we said, this legal amount should actually be 100 times less. It should be a way down, much lower than it was. And we're also going to see shortly that these guidelines were fiddled, changed some years ago as well. In about 2003, they were made more liberal, but we'll come on to that in the more detailed video. But for now, thank you for watching this brief clip.